afternoon here in Spain. It is my pleasure to introduce today's speakers. Igor Weber, a Ph.D. student at the University of Brescia in Italy. With a background in agronomy and entomology from Brazil, Igor is currently focused on developing an innovative modeling framework to better understand and manage Xylella fastidiosa outbreaks in Europe. His research integrates biological processes with environmental drivers such as climate change and farming practices to simulate the dynamics of the disease across diverse conditions. Beyond understanding the system, his work aims to provide a strategic to explore risk scenarios and assess combinations of control strategies, thereby supporting the design of effective integrated pest management schemes. So we now welcome Igor to present his work. Let's go.
Hello. Thank you all for being here, and thank you for the opportunity to present. I'll share my screen. Everything's good. Can we start?
Yeah.
Okay. So I'll present to you the theme of my PhD, a part of my PhD, that is regards the development of a physiologically based approach for modeling Xylella fastidiosa epidemiological dynamics. It is a part of a research that we are developing in the University of Brescia in Italy. That is where we have this group. It is a new research group that develops a lot of interesting works and modeling works in different areas, and we are inside the project, the Big Seal project. We are in charge of developing these frameworks to support the management, and so we will present the first models that we are developing. So mathematical models has been applied to and has been used in different modeling in different systems for modeling and understanding different dynamics such as diseases in humans and also in animals and also in pests to support in some way the management and the understanding of the dynamics involved in the processes that drives the epidemics and the population dynamics. So models has its importance in understanding the system and also supporting us to make decisions. So in the Xylella system, it has been used in the past years, and many models has been used and with different where different aspects of the epidemiology has been explored, and we can mention some of them. So, for example, the study and the understanding of the Xylella fastidiosa epidemiological dynamics, how it evolves in time, how the progression of the disease, so the development of symptoms, how it occurs, the time. And also it's important to understand and estimate parameters that will be then useful for the surveillance. Also, it has been investigated by previous models how vector-host interaction is important to drive the epidemics. So, for example, the preference of the vectors for infected plants, how it is shaped and how it shapes the epidemics. Also, the spatial temporal dynamics of the epidemics in a more local scale, how it evolves, and also in a larger scale, which is also important to predict how the Xylella fastidiosa outbreak will spread. Also, it has been used to predict the global distribution of the Xylella in the world, considering climatic factors, so the climatic suitability of each region and how it is so how the region is suitable to receive and to receive an effective introduction of the Xylella. And it's also is being studied, and models has also been used to investigate and support the optimization and development of more efficient surveillance and outbreak management strategies and plans, as presented by Sendoya in the past seminars, and also investigating all their management strategies such as resistant cultivars and the use of vector control strategies, and how can we define the strategies to manage the spread and prevent the spread and further negative effects of the disease. But in evaluating these models, and it is becoming each time more clear the whole of the local conditions in the epidemiological dynamics. So it is important to consider the environmental component of the disease triangle in the model frameworks, because we know that temperature, for example, is an important factor that drives all the disease triangle. So we have the importance of temperature on the rate of disease progression, on the rate of the bacterial growth inside the plant, and the rate of expression of symptoms. Also, all the effect of temperatures in the vector dynamics and the host plant dynamics. So it is an important component that we can use to improve model accuracy under different components, under different scenarios. Also, another environmental component that can be used, and it is important, is the relative humidity that is very important for the survival, for example, of the vector, mainly during the preimaginal stage, during the egg and nymphal stage. Also, the effect of soil moisture that is important both for the development of symptoms and the progression of symptoms in the plants that is being evaluated mainly for grapevines, but also its effect on the dynamics on the spread and movement dynamics of the vector in the landscape, as it's been observed in the vineyards in California, where the vicinity to areas with water viability, it is more correlated to the spread and the appearance of first infections. So it is also an environmental component that is interesting to be considered in the models to improve its accuracy and its predictability. So we have other environmental components that is important. These are just three of them to mention. And also the importance of considering the vector dynamics into these models. That is, and it is, in most of times, it is simplified in the models that we have, but it's important to consider, for example, vector phenology and its seasonality in the transmission dynamics. So the vector host plant interaction, as we saw, the importance of the quality of the plants in terms of water stress, and how the vector feels this and interacts with these characteristics, so it can shape in some way the dynamics of the disease. Also, the vector movement dynamics within the landscape, and the presence of other hosts of the Xylella in the landscape that can serve as a reservoir. So how can it affect the epidemics? So these are some examples of components of environmental components and other components of the landscape and the system that can be used to improve model realism. So our work inside the Big Seal project and inside the research group that we are part of, it is trying to bring, is focused on bringing biological and ecological realism into the model framework, mainly through the consideration of the physiological responses of vector, bacteria, and host plants to these conditions, to environmental conditions, and also to the interaction between these components, and mainly considering their influence in the Xylella fastidiosa epidemiology as function of these local environmental conditions, such as temperature, soil moisture, and other conditions that we see that is important to be considered. So this approach of improving the realism, of considering and bringing these characteristics to the model can improve the model's accuracy for prediction, system description, and understanding of the system and the effect, the impact of these processes into the dynamics, and also to support the management and the study of and development of management strategies into different conditions, considering exactly this local variability of environmental conditions. So based on this, our objective in this work was to develop mathematical models that describe the Xylella fastidiosa epidemiological dynamics in olive groves. So it is based on a physiologically based compartmental model that I'll present in the next slides. And we have some novelties in this model. That is, we attempt to describe realistically the disease progression dynamics in the olive trees, so based on a physiological approach. We also consider the vector population dynamics and its seasonality into the model, and also the vector host plant interaction dynamics. We modeled it in a very interesting way, and we will show in the next slides. And lately, we applied this model to a case study of the Xylella fastidiosa outbreak in Apulia in Italy. So we applied it to case scenario to study the accuracy and the capacity of the model to reproduce the dynamics that is observed in field. Okay, so starting the model we developed is composed by compartments. So we have the olive grove compartment that is composed by its dynamics. We have the vector compartment, and we have the other vegetative compartments that also are important because they are components of the landscape that we considered. So starting from the olive grove compartment, we represented the different disease stages. So we have initially the plants that are susceptible, so they can acquire the bacteria if there is an infection of these plants. So when a plant becomes infected, it passes to the next stage, that is infected plants, but are plants that has a low bacterial level, and it needs a time to reach a certain level of bacteria, and so it can become infective. So other vectors can feed from this plant and acquire the bacteria and become infected. So for olives, we saw that it is important to consider this phase because of the disease dynamics is a little bit slow in olive plants. So the length of this stage is a little bit it is a little bit long. So we consider this stage to consider this effect of this time between a plant being infected and being effectively effective. So all these three stages are not yet symptomatic. So all these stages are asymptomatic. They don't present symptoms. These two stages are infected and they don't show symptoms. So through develop a disease progression function, these plants that are in the C1 stage, they can start to show symptoms. So we use the classification that is being used in previous studies that are divided into 4 categories. The first category is represents the plants that present less than 25% of its canopy showing desiccation, showing desiccated branches. And then we have the other classes. So we represented 4 classes of symptoms that are either one representing different levels of the branches that are showing symptoms. And the progression from one stage to another stage is represented by disease progression function that we will discuss in the other slides. So this is the basic dynamics that we represented. And the final stage represent the plants that are dead or represent more than 75% of branches desiccated. So are plants that are very heavily diseased. And we also represented the possibility of regression of symptoms. So it is a function that can bring some part of plants that are in a given stage, a given disease stage, to the previous stage. Although it is not well known yet for olive groves, we consider this, we model this, and maybe in future studies with the availability of further studies, we'll be more able to understand how this regression, if this regression occurs, and how it can impact in the dynamics. So these are the olive component. We then have obviously the vector component that interact with these dynamics and is in charge of transmitting and transmitting the dynamics to other plants. So we represented the vector through two main stages. The first is the preimaginal stages that represents the eggs and the nymphal stages. They have a mortality, obviously, so the insects can die by a given function that we'll see. And these insects also emerge into susceptible vectors, so vectors that are not yet infected. And once these vectors interact with the classes of olive plants, if it interacts with a symptomatic or a plant that is infectious, so represented by these red arrows, it becomes infected. So it has an acquisition rate function that we developed, we estimated, that represent this phase of vector that is not yet infected, that becomes infected. And the adult vectors, they contribute to increase the population of preimaginals through a fecundity function. So these are the vectors. And also we represented the other vegetative compartments that are part of the landscape. So we have the herbaceous compartment that represent the ground cover. We have the non-susceptible shrubs and trees that are shrubs and trees that are not susceptible to the Xylella fastidiosa, but can be used by the vectors to feed and to find suitable habitat. And we have the reservoir shrubs and trees, so are species of shrubs and trees that can hold the Xylella fastidiosa and serves as a reservoir of the bacteria to vectors. So we represented a growth dynamics of each of these classes, and also mortality dynamics, senescence dynamics of these classes. And one reservoir plant can become infected if an infected vector feeds from this reservoir plant, so transmits the bacteria to this reservoir plant, and this reservoir plant becomes an infected reservoir plant. So this is the main dynamics, the overview of the model that we developed, and now I'll show in a little more detail some specific interaction and some specific physiologically based functions that we developed, that I imagine this is the main objective of this presentation, is to discuss these physiologically based processes that we represented. So first of all, about the herbaceous dynamics, the function that describes these dynamics is basically a function that has a growth rate and a senescence rate that are dependent on the temperature, the availability of soil, of water in the soil, and also the incident radiation. So we use this model based on a model that was developed by Romero, and it's basically in charge of representing the seasonal dynamics of the biomass. over the year. So here we have day of the year. During spring, we have a maximum availability of biomass of herbaceous, and during summer in the Mediterranean region mainly, we have this reduced availability of biomass because they died due to the lack of water in the soil and also due to high temperatures. We also represented dynamics of the non-susceptible shrubs and trees that we try to represent here the seasonality of the growth of these plants that during the year they produce branches and new leaves, and part of these shrubs and trees that are deciduous, so they can lose some of its leaf during the winter. So we represented this by this senescence function. So it's basically in charge of representing these dynamics that is observed in field. Then the reservoir dynamics is similar to the non-susceptible host shrubs and trees that we observed, but here we have the dynamics of the infection. So we have an infection rate function that I will explain later in the olive growth compartment. And we have also the possibility of reservoir plants that present the infection to recover by any reason to the reservoir, to a susceptible stage that don't show yet don't present anymore the bacteria. Now, about the vector, here we have the functions that describe the vector dynamics. So the preimaginal dynamics, the susceptible vector, and the infected vector equations. We have a fecundity that is dependent on both the susceptible vectors and infected vectors. So all the adults can contribute to the production of new preimaginals. And the idea is to exactly reproduce these dynamics that we see in field. So late in the year, the adults that survive and are available, they lay eggs, and this contributes to increase the availability of preimaginal states. And during the spring in the next year, these eggs, they become nymphs and they then become adults. So they contribute to the increase of adults of adult population. So the fecundity is in charge of increasing this number of insects, of preimaginal insects at the end of the year. And then we have the emergence function that is described by this function that is in charge of making this progression of preimaginal stage to adult stage. We use the model derived from another publication that we did last year that is available in this research, where we have development rate that is dependent on the temperature. So we estimated development rate function that is dependent on temperature, and this is the main function that describes what is the pace of this progression of stage of the progression of the development of the preimaginals to become adults. So it is a temperature dependent function, which is one of the things that I mentioned during the introduction, the importance of considering temperature and environmental factors to describe these dynamics. So here it is one other factor, not one other dynamics that is represented and is dependent on temperature, the rate of development of preimaginals. Then we have the mortality of the vectors of the preimaginal. I'm sorry. So the mortality function is composed by two components. We represented a temperature-dependent component of the mortality. So depending on the mortality, on the temperature of that day, we have higher or lower mortality probabilities of the pre-imaginal individuals. And we also represented, described the density-dependent mortality function, just to avoid that the population would reach higher levels. So we limit the vector population to a level that is related to the carrying capacity of the system. So here the mortality also dependent on the temperature and the density of pre-imaginals. For the adult stage, we have the mortality function that has a component of adult aging. So the adults, as they grow older, they has a probability of dying, to represent this decrease in dynamics that we observe, and also the effect of temperature on its mortality. So also, as we observed in the pre-imaginal stage, we have a mortality of adults that are related to temperature. And for the acquisition, vector acquisition, we have a very interesting function that we described. So a vector can acquire the bacteria if it feeds from infected states, from infected plants. So it can feed from reservoir, infected reservoir, or infected crops that are represented by TY. And it is described by a contact rate function that is described by a mono function that is basically represented in this function. So the rate at which vector feed from a given plant is described and is dependent on the density of the plant. So if we have more plants of a given stage, the probability of a vector that feeding from that plant increases. So it is important to understand and to drive the transmission dynamics. So if you have higher proportion of plants infected, we have higher probability of these insects finding those plants and acquiring the bacteria. So this contact rate function is to represent the dynamics of the vector and the interaction of the vector with the available vegetative compartments in the landscape. So each interaction, each contact between vector and each class is described by this contact rate function that is calculated following this set of equations. Then we have we considered vector preference in the function, in the estimation of this contact rate, to consider the dynamics that is observed in field, that at the beginning of the year, as adults emerge, they leave the ground vegetation and start to go to olive plants, and then during summer, as temperature and water stress is higher, they tend to move to shrubs and trees to find where they find more suitable environment, more suitable microclimatic conditions. So in order to represent these dynamics, we define these preference parameters that are variable in time depending on the time of the year. The vectors present higher preference for herbaceous plants or higher preference for for olive plants. So this is was established in order to represent these dynamics of the vector, the seasonal movement of the vector in the landscape. And the acquisition rate function, so the vector acquisition rate is described by the contact rate and also an acquisition probability that we estimated based on the disease stage. So we assume that the disease stage that are corresponding to higher severity levels of the disease has higher probability of transmitting the bacteria to the vector due to the higher rate of due to the higher bacterial titer that is observed in its plants. But we have on the other side plants that are more severely damaged. They present lower preference by the vectors because we know from literature that plants that show symptoms and show severity symptoms, they tend to be less suitable to vectors. So we have this threshold between preference and the probability of acquiring the bacteria if a vector feed from these plants. So it was represented by this function. And the last stages, the last compartments, we have the olive trees, where the infection function, so a rate at which an olive plant become infected, follows this function, these dynamics, which is basically dependent on the density of infected vectors per plant. So if you have more infected vectors in each plant, the probability that this plant become infected by these infected vectors is higher. So it is described by this function. And the number of vectors, of infected vectors per plant is defined by all that equations that we described in acquisition function. So the dynamics of the vector in choosing, in attacking and feeding from each compartment is important either from the acquisition function as for the infection function. And here we have also other parameters that describe the infection rate function. And about the disease progression, how we model the disease progression, so the rate at which a plant that becomes infected progresses to the next stage. We, due to the long period of each stage, so we know that for the olive groves, a plant that become infected, they start to show symptoms only after 11 months, 10 months. So we needed to consider a delay in these equations. So these equations are described through a delay dynamics. So we have from literature information, or try to understand, to estimate information that could lead us to define what is the period between a plant becoming infected and becoming infectious before showing symptoms, and also what is the period between the plant becoming infected and showing symptoms. We used information, for example, from the EFSA publication that was very important for defining this, and also based on the time between the onset of infection and the death and the complete desiccation of the plant, we estimated the delays of each of these next stage, each of the symptomatic stages. And then we tried to estimate, to calibrate these values using the available data set that we had from field data, to obtain a more reliable values for each of these delays. Now, the disease progression function was considered based was described through a function that was dependent on temperature. So we have the disease progression is composed by three components. We have this first component that describes the accumulation of bacterial growth inside the plant, the component that describes the water stress that this plant are submitted to, and a component that describes the rate of mortality of bacteria inside these plants due to climatic conditions due to temperature, extreme temperature. So this function basically tries to represent these dynamics. So we have this function accumulates bacterial growth during the delay period of a given stage. So if we are in a C1 stage, in a asymptomatic stage, it accumulates bacterial growth as a function of the temperature during all its delay stage. And based on the reached bacterial cumulative bacterial growth rate at the end of this stage, at the end of the delay, we define a higher or lower probability of the plant progress to the next stage. So this was a way of describing describing the dependency of the disease progression in the temperature. The same for the effect of water availability in the disease progression. So we know we consider that an environment that is drier contributes to accelerate the progression of the disease, so accelerates the expression of symptoms. So in the same way, a plant that accumulates more water stress has a higher probability of progressing to the next stage. And for the climatic factor, so for the effect of adverse temperatures, so extreme high or extreme low temperatures, we also consider that if a plant accumulates a lot of climatic stress, a lot of climatic factor during the delay period, it has a higher probability of mortality of the bacteria. So it reduces the probability of a plant progressing to the next stage of the disease. So these are the main equations and the main description of the functions that we considered in the model. And then we applied this equation to this model to a case scenario that is from the province, the Lecce province in Italy, in Apulia region. So it was a data set from 16 olive groves where it was evaluated from 2016 to 2019. The symptoms, the classes of symptoms in each plant, in a lot of plants. So it was evaluated more than 3,000 plants over this period. We used only data from olive groves with susceptible plants. So we considered only susceptible plants, and all these 16 olive groves were very close to each other. So they were very near to each other. So the temperature conditions, the climatic conditions were very similar, and also the landscape composition of these groves was very simplistic. Was mostly composed of olive plants. So if you see around the evaluated grove, they are mostly composed of olive groves. So we didn't have much of the reservoir plants or the non-susceptible shrubs and trees that we considered in the model. They didn't compose a high percentage of the landscape in this data set. So we go to the main results and the final step of the presentation. Here is just for you to see the environment, the climatic conditions of the period. So we have a period, the simulations goes from 2015 to 2019, where to include the dynamics that were observed. So the temperature more or less goes from nearly reached zero during winter, so stays around 10 degrees during winter, and with an average of 30 degrees during summer. And we have here the average daily availability of water in the soil and the precipitation. So we had some years that were very dry in the 2015, 2016, and years that were very wet. So we have different conditions. So the main results here I present only for sites. So we had 16, but we here represent only 4, because just to be more short in the presentation. Here it is the dynamics of the biomass of herbaceous. So we have dynamics that we expected that goes high, that grows during spring and decays during summer. And we have the dynamics of here I represent the reservoir plants. So in gray, we have the total biomass of reservoir that present these dynamics, that is the growth of the biomass during the summer and spring, and the decay during the autumn, and remaining stable during winter. So here we have the biomass that are susceptible, that decays, that goes down during the simulations as the plants become infected. So we have these dynamics here for the reservoir plants that we considered. And for the vectors, we have in this side, in gray, we have the representation of the pre-marginal stage, so with number of individuals per hectare, that present these dynamics, that is, it increases at the end of the year. And then during the beginning of the year, during spring, it decays due to temperature and also due to the emergence of adults. So adults emerge as susceptible vectors, and then they start to acquire, become infected as they feed from infected plants. So here we have the percentage of adults that are infected over the years and the percentage of adults that are susceptible. So we observe that during the middle years, we have most of the insects become infected, and we will understand right now why this happened. So this is mainly because of these curves. So here we have, in the lines represent the simulated dynamics, and the points represent the observed data from the data set that we obtained. So here in these figures, we have the percentage of olives in each disease stage. So we have the percentage of olives in the asymptomatic stage and the percentage of olives in the symptomatic stage are composed by all the 4 symptomatic stages. And we observed that for most of the fields during this intermediary stage, most of the plants become infected. So the vectors find a lot of infected plants as soon as they emerge. So we have that dynamics that most of the vectors become infected during this intermediary phase. So the model could describe with very precise and at least very reasonable precision the dynamics that is observed. And here we observe we have a more detailed dynamics that describes each disease stage. So each symptomatic disease stage. So the colors represent the different symptomatic stages. So in some fields we had more data, in other fields we have just 3 data, but the model could reproduce with a very reliable accuracy the dynamics that is observed in the field. So if we observe, for example, the onset, the start of each stage was very well represented. So the moment that, for example, the C4 stage starts in field was also reproduced by the model, and also the moment that the dynamics has to decrease, it was also reproduced by the model. So for most of the 16 fields that we studied, that we reproduced. The dynamics was very well fitted. Of course, that we have some problems in some parts. For example, here for the fifth site, that it decreases a lot faster the percentage of plants that are not infected and not showing symptoms, as while in field it should stay a little bit longer. But overall, the dynamics was very well described by this model. And here are the cumulative dynamics, cumulative number of plants that we used to optimize the parameters during the model calibration. And further studies that we are developing is a mathematical analysis of this model that we are currently developing. Also, we are developing a Philenus plumarius demographic model. So we firstly developed a phenological model of Philenus, and now we are developing a demographic model, so representing the population dynamics. And we'll present this at the 28th Italian National Congress of Entomology in June. And also we are developing a very interesting model of the disease progression of the Pierce disease in grapevines that we will present also in June at the conference of the European Foundation for Plant Pathology in Sweden. So this is our last model that we are currently developing, and we hope to bring some interesting results in the next months. So I would like to thank, of course, everyone that contributed directly to the model, but also all the experts and the research groups that contributed either with data and with information, and especially to the Big Seal project, the financial support, but also the project support from Blanca Landa, from Ana Sciences, that is very important for the development of these models, mainly because of the reduced availability of data, of information. This project is very important because it brings us very new and fresh data to support these models. So thank you all for the patience, and I think I hope it was at least clear the idea of the model and the idea that we try to represent here. And thank you all.
Thank you, Igor. Yes, for sure, it is a very interesting model, and now today I learned a lot from you, and I think it could be very helpful for my future project. So we'll now open the floor to questions from the audience. So if you have any question, please feel free to raise your hand or directly unmute yourself.